Hello, everyone, and welcome to this week's edition of the Extracellular Vesicle Club. My name is Kenneth Whitwer, and I'm the Chair of Science and Meetings for ICEV. And I'm happy to welcome Tanina Arab and Emily Mallick to give this presentation on an article that we finally published in Journal of Extracellular Vesicles in April of this year. This session today is actually going to be the first of a little mini-series that we're having on single particle analysis. So we will have several more presentations over the next few months that will feature articles on single particle analysis. Tanina Arab is a postdoc at Johns Hopkins University, and Emily Mallick was with us as a research technologist, and she has since moved to GlaxoSmithKline. So Tanina, Emily, welcome. Welcome back, Emily. It's great to have you here today, and I'm going to hand the reins over to you.
All right. Thank you, Dr. Whitwer and Tanina, for having me back to present this paper with you guys today. So I'm going to go through, and Tanina's going to take us through the second half of our paper titled Characterization of Extracellular Vesicles and Synthetic Nanoparticles with 4 Orthogonal Single Particle Analysis Platforms. Now, as most of you are aware of the different EV isolation and characterization methods, I'm just going to briefly touch upon our experimental design and the 4 technologies that we have investigated. So as input samples for our 4 platforms, we used both artificial particles and biological material. We chose to use both silica and polystyrene particles because they have known diameters and compositions as well as distinct refractive indices. So the silica particles that we used were purchased pre-mixed at 4 distinct sizes, where the polystyrene spheres were purchased separately and then mixed at equal concentrations of each size. On the biological material side, we used EVs that were isolated from 2 well-characterized cell lines with unique tetraspanin profiles. So our first cell line was the human H9 T lymphocytic cells, which are high in CD81 and low in CD63, while alternatively, our human U937 promonocytic cells are low in CD81 and high in CD63. So these unique profiles were able to help us quite extensively when it came to utilizing the fluorescent features of the platforms and then with the single particle analysis of things. So just briefly, we used conditioned culture media from both cell lines that was isolated by either a combination of ultrafiltration and size exclusion chromatography using 70 nanometer columns or by differential ultracentrifugation. Now, the products of these isolation methods will be referred to as either SEC EVs or 100K EVs throughout the presentation. So before beginning our characterization using the 4 different platforms, we wanted to ensure that we had high quality input samples. Therefore, we used common characterization methods such as Western blot and transmission electron microscopy. First, Western blot revealed characteristic cellular CD81 and CD63 expression patterns from our cell types, as I previously mentioned. The signal for the SEC EVs tends to be slightly stronger in comparison to the 100K EVs. Now, interestingly, when comparing our Western result from the cell lysate to the EVs themselves, it appears that CD81 is enriched in the H9 EVs, but not so much in the cell lysate, whereas CD63 is enriched in the U937 cell lysate, but not so much the EVs. Finally, calnexin is mainly present in the cell lysate, with very little signal being transferred into the EV fractions, which is what we would expect. Now moving to TEM images, where we revealed the presence of the cup-shaped particles, which is the characteristic EV shape post-fixation on the grid. The 100K EVs tend to display a higher background and apparent non-EV particles as compared to our SEC fractions, which we suspect is due to the presence of protein aggregation that has been co-isolated in the centrifugation process. We were also able to confirm the size and purity of the silica and polystyrene spheres at the bottom of the screen. You can see that a variety of sizes are seen in these images, indicating that we did have 4 populations as a homogeneous mixture. Okay, so as you're all aware, the nuts and bolts of our paper came from these 4 orthogonal technologies from which we assessed sizing, counting, and phenotyping when available of our input samples. So I'm just going to briefly touch upon the 4 different technologies so that we're all on the same page. The first that we used was single particle interferometric reflectance imaging sensor, or SP-Iris, which is essentially a platform that captures the particles onto a chip by affinity reagents, which in our case were antibodies, to surface antigens. After these particles are imaged for sizing and counting, then fluorescence detection may be done for up to 3 channels for surface antigens or internal molecules following fixation and permeabilization protocol. Our second was nanoparticle tracking analysis, or NTA, which is an optical method to track single particles and assign size and counts. Using Brownian motion, we can calculate the hydrodynamic sphere equivalent radius of each tracked particle. And then additionally, fluorescence filters can be used for detection of fluorescent channels. Third, we have our microfluidic resistive pulse sensing, or MRPS, which counts and sizes particles as they pass through a pore between microfluidic chambers. Now, occlusion of the pore results in a measurable change in electrical signal, which defines an event, and this event is proportional to the volume of the particle. Now, for this technique, we use different disposable cartridge pore sizes to detect particle populations within different size ranges. As a non-optical technology, fluorescence detection is not available for this instrument, which is making it is unique in our grouping of 4 here. That is the only one that's not capable of this fluorescent detection. Now finally, nanoflow cytometry, or nano-FCM, is a flow-based technique that detects nanosized particles by scatter and or fluorescence. Now, compared with traditional flow, a smaller flow channel reduces background signal and lowers system pressure, increases dwell time of the particles for enhanced signal integration. All right, now with the introduction taken care of, I am going to hop into our data. So I'm going to begin with our silica sphere size distribution. So as I mentioned, we purchased pre-mixed silica spheres that had nominal values here of 68, 91, 113, and 151 nanometers. These values are indicated by the vertical dotted lines on each graph for visualization purposes, and the exact concentration of this particle mixture was unknown. So beginning with SP-Iris, 4 distinct populations were detected with diameter modes around 70, 100, 120, and 150 nanometers. Moving to NTA, which detected a broad population with a diameter mode of around 105 nanometers. MRPS could resolve 4 distinct peaks for each individual cartridge, which is depicted here by this inset in the upper right-hand corner of the graph. However, this distinction was somewhat masked when averaging the 3 replicates, ending us with a little bit of a wonky-looking graph here. We speculate that this may have been due to aggregation of silica spheres when using PBS as a buffer. Finally, nano-FCM also was able to resolve 4 populations with distinct peaks at diameters of approximately 66, 85, 112, and 154 nanometers. Now, these peaks are almost identical to the nominal values, which we believe is because these beads are the exact ones used to calibrate this instrument. So it's exactly what we would expect. I'm going to switch now to our polystyrene. Now, in order to replicate the silica spheres to the best of our ability, given the available reagents, we mixed polystyrene spheres with nominal diameters of 70, 90, 125, and 150 nanometers to a concentration of 1 e to the 12th particles per mil. And that's what we use as our stock solution. Now, SP-Iris again identified 4 distinct populations with diameter modes around 80, 110, 140, and 170 nanometers. This size overestimation is likely attributed to the higher refractive index of polystyrene in comparison to silica, although the 4 peaks were resolved. Again, NTA detected a broad population with a diameter mode of around 105 nanometers. MRPS could resolve 4 distinct peaks at diameter modes of 71, 92, 123, and 150 nanometers, even after averaging the 3 replicates this time. Now, Although a calibration step is not necessary for the MRPS before use, if the user would choose to do one, polystyrene nanoparticles are recommended for use, which is why we believe that the resolution was so clear here. And then finally, for nano FCM, again, resolved 4 distinct peaks at 85, 120, 170, and 225 nanometers, with a possible fifth smaller population residing right around here at 60 nanometers. So we wanted to include this slide just to indicate that we did measure using the MRPS and the nano FCM, both of the polystyrene and silica nanoparticles at different dilutions. So again, we just want to point out here that regardless of the dilution, the readouts were pretty much the same, and you can see much clearer resolution with the polystyrenes for the MRPS and the silica for the nano FCM, which was consistent with our previous findings in our main figures of the paper. Now, because NTA did not appear to resolve our 4 different bead populations, we assessed the polystyrene particles at their individual sizes before they were premixed to try to resolve each individual population. As you can see here in this graph that was not shown in the original paper, when looking at the larger populations of 125 nanometers and 150 nanometers, the mean diameter was quite comparable to the theoretical diameter, only ranging a slight bit. However, this was observed to a lesser extent when comparing the smaller diameters at the 70 and 90 nanometer range. Now, overall, though, in comparison to the other instruments tested, NTA was still only able to detect that single broad peak using our mixed beads around 124. Okay, now going from our size distribution to quantification of our nanoparticles. So for SB Iris, our data is plotted independently of the other 3 platforms just due to its inability to directly provide quantification values in terms of particles per mil, but it rather provides a particle count dependent on the number of particles that are captured on that specific chip. So for these experiments, what we did is we simply added a fixed volume of particles to each chip and allowed the solution to dry. We would not expect these particles to be bound to the chip by an antibody-antigen interaction. Therefore, we did not include any washing steps before scanning them, which was something that was standard for the biological materials. Now, for the silica particles, a mean of around 3,000 particles were detected per antibody printed spot, with no appreciable differences between groups, i.e. groups 1 through 4. There was, however, a considerable range of particles detected within each group, ranging between about 2,000 and 4,500 particles per spot. Now, quite similar results were obtained for the polystyrene spheres. There were no differences between groups, but again, events ranged from about 1,000 to 3,000 within groups. So based on our nominal polystyrene bead concentration and the surface area of the chips, we were able to calculate that approximately 10,000 particles per spot were to be expected, which is depicted up here by our horizontal line. It is possible, however, that the beads may not have dried regularly onto the chip, or they could have even been repelled by the antibody printed surfaces, which is why our results were so much lower than expected. Now, following interferometric measurements, our chips were probed with 3 fluorescently labeled antibodies against the 3 tetraspanins, our CD81, CD63, and CD9. And this was just to assess background binding. As you can see, background binding was quite negligible for both the silica and polystyrene particles, with a little bit of CD9 popping up here in the silica beads, and a little bit of CD63 popping up in the polystyrene. But nonetheless, these events do not exceed about 1,000, so we could just consider those background noise. Now for the quantification of our particles assessed by NTA, MRPS, and Nano FCM. So for our silica particles, MRPS estimated a concentration that was about 10 times higher than that of NTA, and Nano FCM reported a concentration that was somewhere in the middle, approximately 3 times less than MRPS. It should be noted, though, that all 3 replicates from each instrument were precise, although not accurately measured at the same concentration. But then again, we didn't really have an accurate concentration to begin with, so it's kind of hard to compare there. But for polystyrene, all instruments were both precise and accurate. So again, we do attribute some of these differences between the silica and polystyrene particles to the refractive indices of each particles, which are listed here. Silica being around 142 and polystyrene 159. So at this point, I'm going to hand it over to Tanina, and she's going to take us through the biological materials.
Thank you so much, Emily, for presenting our data on the, let's see here. Next slide, please. For presenting our data on the artificial spheres, I'm going to take care of the second part, which is the biological samples. So EV preparation from H9 and U937, as already mentioned, were measured for all these platforms. And for H9, as you can see in this graph, SPIRS returned almost identical size distribution profile for both enrichment methods. Next, please. In contrast, NTA, MRPS, and NanoFCM returned data indicating particles that are slightly smaller in diameters for the 100K EVs comparing with SEC EVs, although we can see similar particle size distribution overall. Next slide, please. So for U937, sorry, I'm a bit lost with this screen. For U937 derived material, similar pattern were observed with SPIRS and NTA, detecting more particles at smaller diameter from the 100K EVs compared to the SEC EVs, but again, with similar particle size distribution. And MRPS, which is in the bottom left panel, produces equivalent particle size distribution and particle number between the two enrichment technique. Next, please. This is in contrast with NanoFCM detecting a higher particle count of smaller particle diameter for the size exclusion chromatography EVs comparing to the 100K EVs. With this time, the size distribution being significantly different. Now, for all sizing measurement, there is a substantial variation between replicates, which limit our conclusion. And this is indeed a little bit crowded, especially for NanoFCM. Moreover, we know that EVs are highly polydisperse and therefore will naturally lead to greater coefficient of variation and different reflection indexes. So this make conclusion a little bit harder. Overall, though, the results are broadly consistent with the expectation that UC pellet may contain non-EV extracellular vesicous particles, which are around the same size as EVs. Next, please. However, we would like to be careful because evidence of aggregation by UC is not apparent in our data that we are presenting here. And for the next, please. Okay, you already switched it. For the obtain, go back in the previous slide, please. Sorry. So for the obtained data for H9 and U937, it does seem like NTA is capable of detecting shifts in population distribution. And this capability actually may be more important for biological particles rather than being able to resolve artificial sphere subpopulation. Next, please. So after size distribution, we proceed with particle count for biological samples. And consistent with expectation based on cellular tetraspanin expression and release that Emily already presented in Western blot experiment, SPIRS is showing that H9 particles were captured by CD81. So these are the blue round shape, and this can also express CD63. When for U937, which is the right panel, it seems that there is capture with CD63, but no capture with CD81, which is comparable to the isotype control. Next, please. All right, so for the three remaining platforms, several trends were seen. So overall, this data were consistent with the result for silica spheres, where counting in the NTA was lower than the counting, so this is the pink color, was lower than counting with NanoFCM, which is the yellow color, and this one is lower than MRPS, which is the green color. And this is correct for both SEC EVs and 100K EVs. After taking into account dilution factors, MRPS and NanoFCM seems to measure lower particle concentration for size exclusion EVs, size exclusion chromatography EVs comparing to 100K EVs, and likely this is due to the depletion of protein aggregation in SEC EVs. And this is kind of interesting when comparing to the data we just seen for the polystyrene spheres, where all these techniques returned similar values. But we can clearly see here that for the biological sample, it's not the case. Next, please. Next, we were interested in buffer and controls as part of rigor effort we put in this study. So upon review or request, we wanted to address if low concentration of twin detergent affect MRPS measurement. And so the hypothesis here was, what if the presence of twin in our buffer when using MRPS could lyse EVs, and therefore this is why we're seeing an increase in particle concentration with MRPS. So this is the actually results from Edith Bouzache lab, where they compared different detergent with different EV subpopulation, including apoptotic bodies, microvesicles, and exosome, and these are coming from different cell line. So basically, they subjected these EVs to different concentration of a reagent that are included but are not limited to twin. And I am encouraging everyone to have a look in this paper if you didn't saw it before. But mainly their conclusion about the percentage of twin is for apoptotic bodies, 10% of twin is needed to induce these EVs to be lysed, similarly to microvesicles. And for exosome, it's even higher, 50% of twin is needed. And this is way above the concentration that we use, which is 0.5%. Next, please. Nevertheless, we wanted to test our archived samples, and therefore we suspend EVs in different percentage of twin 20, ranging from 0.1% to 0.9% before subjecting them to MRPS measurement. The graph we are showing here is a presentation of results obtained for H9 and U937 side by side. And as you can see, across tested detergent dilution, measured EVs concentration average about 1.7 10 to the 11 for H9 and 1.5 10 to the 11 for U937, with no real correlation between count and detergent concentration. So we can conclude that indeed the presence of a very low percentage of twin in the MRPS buffer doesn't hurt the particle concentration afterwards. Next, please. So though we observed some variability in the size profile, but again, there was no evidence of a clear decrease in particle size, which would suggest that indeed we were able to lyse these EVs. Next, please. After counting these EVs from the biological samples, we phenotype them, and fluorescence measurement for biological particles were done with 3 platforms. As Emily already mentioned, we don't have the option to do fluorescence investigation using MRPS. Therefore, we restricted this work to SPI-RES, NTA, and nano-FCM. So I'm going to present the SPI-RES data separately for one reason. I mean, one particular reason is because this is a capture-based method, and therefore it doesn't take into account bulk EVs in contrast with other platforms, but it does take into account only the captured one. So this data are the label-free. So in the label-free count, SPI-RES platform measured individual particles by antibodies, and that's putatively positive for one antigen. So it's already a single particle phenotyping platform without even adding a fluorescence labeling. However, we did fluorescence detection with CD81 and CD63. So basically what you see here is that we capture with CD81, CD63, and isotype control, and we labeled with CD81 and CD63. In panel A and B, we represent a result from chips incubated with H9 SEC EVs and H9 100K EVs, respectively. And you can see that the EVs captured by CD81 antibodies were also generally positive for CD81 by fluorescence. So this is the round shape dark blue. And some were also appearing to be CD63 positive, and these are the light blue or round shape. In contrast, CD63 capture spots were largely lacking fluorescence for H9 EVs. We already saw with several results that these EVs, from H9 do not express CD63 highly. The bottom panels, C and D, display results from chip incubated with U937 EVs. And as you can see, events on CD63 capture spots were also positive for CD63 by fluorescence. So these are the light blue round shape, and CD81 linked fluorescence was at the background level for all spots with U937 EVs, and this is also consistent with what we saw in Western blot results. So not that the number of positive events are higher in fluorescence mode than with label-free imaging. So here we have, like in D panel, almost 5K events, and it can reach 10K events in the 8th panel, when previously in the label count, we were around 3K events. So this is likely because fluorescence detection is more sensitive than reflectance imaging, which make this this platform suitable for multiplexing and fluorescence. Also of note, and this is also some observation that we are seeing not only with this experiment, but also with other studies, is capturing EVs onto the chip via surface marker can make those markers in the EV surface less available for subsequent binding but fluorescently labeled antibodies. So for example, I will ask you to focus with me on panel A and the light blue dots. So CD81 captured particles that also display CD63 appear to have more CD63 available for binding by fluorescence antibody, comparing to CD63 positive particles. These are the light blue round shape. which seems to have less available CD63 because it's already sequestrated by surface binding. So we thought that this is a good observation to share with you today. And those phenomena are observed independently with the method that we applied. So it's correct for sec EVs and 100K EVs. Next, please. So similarly, as a part of our rigor effort, and conditioned media subjected to the same process used for EV separation was assessed for particle count and fluorescence using SPIRS. In label-free capture, SPIRS recorded only several hundreds events per spot, when in our EV samples, these were a few hundreds. And next, please. So in fluorescence mode, the background level is even lower, and it's approximately like tens of events. And we have observed no difference between tetraspanine and isotype control, or between preparation by differential centrifugation or size exclusion chromatography. So for the two remaining platforms with fluorescence capabilities, respectively NTA and nano-FCM, results are shown as percent of total particles that were stained comparing to the original sample. So let's start with H9 in the upper panel, the upper left panel. There is approximately 40% to 50% of detected particles from H9 cells that were positive for CD81, according to NTA. And these are the round shape, the pink round shape, while a little to no CD81 signal was detected for U937, and these are the bottom panels. CD63 was not detectable with NTA, even when expected, since we expected CD63 in U937 material. In contrast, though, nano-FCM, which are the yellow round-shaped colors, detected either CD81 or CD63 in a small percentage of particles. So for H9-derived particles, which is again the upper panel, within the same methods. So if we focus only on H9-seq EVs, we can see that there is no significant difference between CD81 and CD63 signal with nano-FCM, and this is also true when we have a look on the F panel, comparing CD81 and CD63 measured with nano-FCM in H9 100K EVs. So taking together, this data are not showing a real major difference between SEC and 100K separation methods. Next, please. All right, so this brings us to our summary conclusion slide. So for any platform and configuration, particle count is accurate only within a certain range, and sensitivity of particles of different sizes and refractive indexes should be considered, since EVs are polydispersed and therefore will have heterogeneous dynamic light scattering. We also showed you that the different size population within a mixture of synthetic nanoparticles can be identified by SPIRS, MRPS, and nano-FCM, but not in our hand, at least by NTA with the default setting. Next, please. So SPIRS, NTA, and nano-FCM have fluorescence particle phenotyping capabilities, while MRPS does not. And multiplexing biological particles phenotyping for tetraspanine is easily achievable with the SPIRS platform, since for one capture antibody, up to 3 antibodies can be used for fluorescence detection. Nano-FCM theoretically has multiplexing capabilities that we didn't investigate in this paper, And to some extent, NTA can be used with different lasers. However, this requires switching filters, and it may be challenging to follow or track the same particles in different channels. Next, please. So thankfully, there is some useful biological reference material that are now available in the market since this paper have been published. And we certainly need more of those with known diameters, known refractive indexes, and behavior in different solution buffer that we are all using in our experiment. Therefore, we can conduct a more strong investigation about single particle analysis as well as multiplexing configurations. Next, please. So in the same theme, actually, to strengthen conclusions and study, one may consider orthogonal technologies, of course, when possible, as opposed to relying only to data outcome from single platform. Next, please. So not only acquisition costs, but also recurring ones should be considered before choosing any platform. And last but not least, we wanted to stress that none of the challenged platform tested here is better or worse. It is up to the user to make the most out of each platform with regards to including, of course, proper control. Next, please. So more detailed summary table has been included in the paper with size and particle detection ranges, among other valuable information that readers can find. So please have a look on the paper. Next, please. So with that, we would like to thank you for your attention and for attending this journal club session. And thank you everyone that contributed to this work, all the Whitwell Lab members, former and present. Special thanks to Emily. I'm really happy to see you back for this journal club and presenting the paper with me. And all our collaborators at Johns Hopkins University and beyond, company representative for constructive feedback and discussion along the way, and of course our funding agencies for supporting our work. And we're happy to take any question you may have. Thank you.
Nicely done, Tanina and Emily. I really appreciated your presentation today, and it looks like we do have some questions now in the chat box. So let's go ahead and get to them. We will. So I noticed that a lot of the questions have to do with settings and technical details about the experiments. And I just want to stress that a lot of these experiments were done 1.5 to 2 years ago. So it might not be immediately on the minds of our presenters. So I have put the link to the paper and then also the link to our EV track and our flow repository details in the chat box. So you're welcome to consult with those for anything that we're not able to answer, off the top of our heads here. So let's start though. We'll start with first of all, me doing what I'm supposed to do, which is to allow people to unmute themselves. So you should be able to unmute yourself now. So what I'm going to do is just read these questions. Emily and Tanina can answer them. And then if there's a follow-up, feel free to unmute yourself and jump in. So we have a question here from F. Masajo, who asks, how is your buffer looking with these 4 methods? So I guess, what are the differences in buffer composition that you use for these different platforms? And some of this I think you've addressed in the meantime, but go ahead and tell us a little bit about what some of the what some of the characteristics of buffers might be that would be important to the outcome of experiments.
I can take this one. So basically what we have been using for our EVs was mainly PBS with protein inhibitor and phosphatase inhibitor when needed, especially for Western blot experiment. But for platforms, it was mainly PBS, and only for MRPS that upon recommendation from the manufacturer, actually, we had to add 0.5% of twin. And also for the SPI res, we were stick to what was available on the kit. So solution A and solution B, which were not 100% disclosed in term of composition, but it was also mainly PBS and maybe a very, very low percentage of detergent on it. Do you have anything to add to that, Emily? Please feel free to jump in.
Well, so the questioner has followed up to say that what they really meant was, what do the plots look like when you run buffer alone?
Oh, I see. So
yeah,
actually,
I didn't get that. Sorry.
No worries. So for NTA, it was below the detection limit because for NTA, you have to have at least like 200 particles in the field of view to be able to, at the end, like for the software, to be able to calculate back your concentration. So basically, we just generated this PDF with non-particles that were able to be counted. And for SPI-RES, I think we already showed the data of our controls. Events were definitely lower in the condition media and the buffer comparing to our samples. For nano FCM, it was around 10 to the 6, I believe, particle per ml. And for MRPS, Emily?
Yeah, I can't remember that off the top of my head, but it was definitely negligible in comparison to the samples. It didn't look anywhere near. I think it might have been 10 to the 6th, 10 to the 7th as well.
Yeah, so there is a background that you're going to find for any of these platforms, but it's going to be well below the biological or the spiked in synthetic materials. And it's of course going to depend too on what exactly that buffer control is. So a PBS is one thing, but if we're using let's say we're using cell culture conditioned medium, if we just take the medium without conditioning, there might be other factors in there that are going to cause a higher background than what we'd see in the buffer alone. So that's pretty clear. So thanks for the question on that. Now we have a question about the data presentation. Amber Murray would like to understand whether why do we use mode in one case versus median or mean in presenting these data?
So I guess this is referring to artificial spheres. We have been saying a lot mud, just because we were, so with artificial spheres, we mixed known particles with known diameters. So we expected the mud, which is the most represented, which is the most present population in a given sample, to give us back those nominal values. So maybe that's why today during our talk we were saying a lot mud and rather than mean, just to have this signal out of the background. Yeah, I don't know if this answers the question.
Yeah, I think when you have those 4 different populations or 5 or however many you're measuring, you've got to draw a line somewhere and say, what is the most common value in here? And so we chose the mode to present that, but or to represent that, but it could just as well be some other measure of the the average generically. So median or mean would also give you a fairly similar answer. Okay, then a question from Alyssa Calder. What was your dilution of sample for the Zeta View NTA? How much sample did you load? So I think that, well, I'll let you answer that.
All right, so for NTA, you need at least a strand of 1 ml to be able to inject all your samples and to be able to feel the capillary and to feel all the instrument and to have like accurate detection. And we have been so using dilutions. We started with the most concentrated samples to the lower one, and we stopped when we had this roughly 200 particles per field of view. If I remember correctly, it was something around 1 to the 100 in terms of dilution.
Yeah, so you might want to take a look too at the table that was at the end of our presentation. And at I think some of the other papers that we're going to be hearing about in coming weeks, to see what the ranges are, the generally accepted ranges of what each of these instruments can measure. And these are, of course, also going to be in turn dependent on the exact settings that you use. So it's just important to make sure that you're within that range when you're measuring anything on any of the platforms. And okay, so Yvonne Couch has sent a message, and she doesn't have her mic, but then I see she has two questions, and Gao also has a question that's similar to hers. So her first question is, has anyone done something similar with synthetic liposomes as controls rather than these other materials? And then she asks later, along with Gao, what about blood, like plasma or serum? Have we tried that on these platforms? So let me tackle the first one. Because I know that there have been a few papers in the literature on liposomes. And so one of them is this paper that came out from Adriele Prinamello that appeared in JEV a little bit earlier this year. And so that's a paper that we're just trying to find a suitable time for them to present, but that paper will be presented here in an upcoming session. So they did a libosome population that was somewhere a little bit smaller than 100 nanometers in diameter. I don't recall exactly what that was. There was another paper in Journal of Pharmaceutical Sciences a couple of years ago where they did this with liposomes and MRPS. And then we also had an EV club last year with Wyatt Vreeland, who's at the National Institute of Standards and Technology in the US. And so Wyatt shared with us some of his data where he had made some liposome standards in very defined diameters, and he ran those through his asymmetric flow field flow fractionation instrument to separate those and characterize those. So those are some of the things that have been done with liposomes. And so then I'll hand it over to you, Tanina and Emily. What about blood products? What about these complex biological fluids?
So the only comment that I have to add to that is one of our collaborators from the Pienta lab, they did some testing from urine and plasma samples on some of these platforms. That is what's coming to mind. Tanina, do you have anything to add here?
So what was the question, if there is any study that compared plasma samples across platform? Is that the question?
Yes, essentially, yeah.
Okay.
Because of their complexity. So there's so much other stuff in there compared with what we used.
Yeah, not from the top of my head actually, but definitely there is papers that were using plasma with these platforms, but across different papers and different studies. So if this is different, I mean, if the question is it doable? The answer is yes. Where is the most suitable platform for plasma? I don't know. And I really don't have a paper in mind that compared platform using plasma. Do you, Ken?
Well, we've done some of that work where but we always do some form of separation before we would look at the material in plasma, just because the EVs are at a very small minority in plasma compared with lipoprotein particles and other extracellular particles that are in much higher concentrations, like orders of magnitude higher. So it's difficult to get, unless you're able to do phenotyping, it's difficult to really understand what those different populations are if you go with an unseparated and especially an undiluted biofluid. Now, it may be possible to dilute to the point where you're able to count things. But again, you would have to do some form of phenotyping to understand what were EVs and what were not. So it's definitely much of quite a challenge still. Although there is, if you can be specific, if you can capture, if you can somehow segregate the EVs away from other parts, it might be possible. So let's now go to Karen Boussard, who asks, were polypropylene beads also tried? So if you, we did polystyrene, we did silica, but what about polypropylene? So why did we choose these two particular nanoparticle types?
Basically, the silica spheres, we had them already available on because these are used for nano FCM calibration, and we were thinking about artificial spheres with the higher refractive indexes to see what the outcome will be when comparing two different artificial particles that have different refractive indexes. But is there any other reason, Emily? Because you started this work even before I joined the lab. Is there any other reason why you went to that?
It's funny actually. I think the suggestion actually came from Dr. Whitward to use polystyrene as well. But another reason to advocate for the polystyrene is because if you would choose to do that calibration step for the MRPS, you would use polystyrene. So it kind of ended up being comparable since it was like kind of a calibration for each one of the instruments. There was some sort of comparison that could be done there too.
Yeah, so I would just add to that that there are a lot of synthetic particles that you could use. Even if we start talking about liposomes, we can have liposomes of different composition, different sizes, and we know we happen to have these materials in the lab, and we know a lot about them. So these have been characterized to an inch of their lives. They're traceable standards, and so we have something to compare with there. Of course, they're not meant to be a substitute or similar to the EVs that we're measuring. The refractive index of the silica spheres is closer to EVs than that of polystyrene, but it's still not the same. So that is certainly a caveat here. But we also made an easy target, I guess, out of some of these things. So can we actually separate or distinguish these different populations of particles that we know a lot about? All right, let's see what we have here. So Amber Murray is asking about the NTA settings that we used and how did we choose those settings? And then also where we suspected protein aggregation, did we confirm that?
If you have anything to add about NTA, I could take some of the protein aggregation.
All right, okay. So actually, basically, all the instrument, we didn't want to play a lot with the settings, and we used mainly the default settings because, of course, if you start playing with the settings for each instrument, you may introduce bias that will be out of control afterwards to draw conclusions. So we didn't play that much with the settings on instrument, except for the ones, of course, we were just following the protocols. The ones that needed calibration, we calibrated them. The ones that didn't need any calibration, we just used them as the default settings.
And then to touch upon the protein aggregation, I guess we would go back to our characterization by Western and TEM, and that was essentially the extent to which we looked at what protein could be included there. We didn't do any other type of tests, from my knowledge, to determine whether or not we had additional protein in there or if we could degrade any and remeasure any type of samples. Does that sound correct, Tanina?
Yeah, that's correct. We basically used calnexin and PEEP, which are non-EV markers for Western blots, and we also rely a lot on the background seen in the TEM images to suspect the presence of protein aggregation in UC samples. But again, we are very careful about that because we didn't push it and investigated it to show how these aggregation are comparable to EVs, or at which ratio, for instance, they are present comparing to EVs. So, yeah.
Yeah, if you were to measure the protein concentration of your EV preparations and compare it with the particle counts, you would see fairly large differences between what is obtained with an ultracentrifuge versus what we would get with size exclusion chromatography. And then, of course, the aggregation potential of those proteins is also different depending on the properties of the buffer that things are in. So there's, I think that there's a evidence that was not included in this paper, but we do have a pretty good idea of the differences between the separation methods in terms of how much protein is present or is purified away. As for, I did just want to say one other thing about the settings. So I guess a kind of dirty secret in our field is that if you have a concentration that you want to get, you can somehow change your settings to get that concentration. And so that's why we want to be very careful that if we are making comparisons, even with the same instrument on different times, it is really a good idea to include some reference material there, to kind of understand how the performance of the instrument is even varying between runs or between operators. So keep in mind there that there's a real need for the individual lab to standardize how they run the instrument, and then also if we're trying to compare between labs and between instruments, to make sure that we are really comparing back to something that everyone can use, if we want to make that comparison. All right, let's see what other questions we have here. We have a question about, here we have one from Lilia Panushkina, who asks, have you tried to play with the raw NTA data? So yes, I believe that we did. And Tanina, so you can tell us a little bit more about what we tried to do there to see if we could get some different populations.
Yeah, we tried different software features with the NTA software. Actually, this is the slide that Emily showed with the gates. And we tried to separate these spheres. Yeah, thank you, Emily. So this would be slide 8. These are the right panels where we where we reopened the data generated with polystyrene spheres and silica spheres in the NTA software, and we tried to assign gates based on intensities. But to assess just the possibility that the individual bit size could be resolved. And for polystyrene bits, the most intense signal is slightly toward larger return size. However, there is no apparent really size or distribution differences between the medium and low intensities. And just as a reminder, we have 4 population here, and really we're not able to separate these 4 populations. Yeah, I don't know if this is what Lilia was meaning by playing.
Yeah, if I recall correctly, Lilia, you had asked us about this, and we did this experiment in part because of what you said. So
yeah, thanks. If I just commit, if I see the data like everybody of us that we don't see any like distinguished population, the first thing coming to my mind that is probably the data being pre-analyzed in a way that they just an approximation, right? And it might be that the real data that been obtained from the mixture were actually distinguished. But that is just, that's why I've asked about the raw data, and not inside the software maybe, but just taking TXT file and so on. But yeah, I get your point that you basically were not able to see it, but because we actually had more like the same problem. And I'm just really wondering, and representation of the data from the different platforms could be also not ideal, and maybe also keep it in mind and just jump into some raw data. But yeah, thank you.
Indeed. So we have a question from Pia, who asks, how were the silica and PS beads captured to the SPRS platform? Great question. So we couldn't, obviously, because they didn't have anything that we could grab, with an antibody. So we just dried them onto the surface. And so this is a caveat of that experiment, that they could have had uneven drying. So if you look at the results, they're actually quite close to the nominal values. So it's a little bit lower, but that could just be explained by the uneven drying. So, I think sometimes with some of these techniques, if we get within tenfold of of what we expect, it's pretty good. And in this case, we were a little bit lower, but it looked pretty good with the counts, but excellent really with the sizes that were assigned by that platform. And final question from Aslan Deghani, who asks, did you consider comparing the concentration of the silica beads with that of those monodisperse particles or their mixtures that we use? So we knew what the concentration of the PS beads was. So could we take that and use it to kind of calibrate for what the silica beads or the EVs were?
I'm sorry, if you don't mind, I can explain it a little better. Sorry, it's hard to
Yes.
track it down. What I meant was that, so for silica beads, you didn't know the theoretical concentration. However, if you measure the concentration of monodispersed particles, each size of these particles, and then you know a detected concentration for each of them, and depending on how, let's say, equally 1-to-1 ratio you are mixing them, you should know, you would be able to know the concentration in the mixture. However, then when
sorry, you mean for the silica?
For example, yes.
Yeah, the silica
so
we already purchased pre-mixed. We didn't mix them ourselves.
So, so you wouldn't be able to measure them in a monodispersed sample separately.
No.
Because I was wondering, because masking effect is a known phenomena for NTA, that larger particles tend to mask smaller particles. And I was wondering, possibly one of the reasons that you observed a difference was because of masking effect, but what if there is some weird phenomena happening in the other two techniques leading to, you know, significantly higher concentration as well?
Yeah, so for our polystyrene populations, we did know the concentrations of each individual population there and the sizes. But for the silica in this particular instance, we didn't know that. So we don't have those individual populations. Rather, we just had the mixture. All right, well, I believe that we've come to the end of our questions, and we've also reached the end of our hour. We've gone a little bit over. Thank you, everybody, for attending today. It's been good to talk with everyone about these results. And like I said, we're going to have several more papers comparing platforms that will be coming up in the next few months. So stay tuned for those. And I'd just like to thank Emily Mallick and Tanina Arab for sharing with us today. And I hope to see all of you at a future EV Club event very soon. Take care, everyone. Hope you have a great rest of the week. Bye now.